Hi, today we are going to talk about the brain, also known as the encephalon. The word encephalon comes from the Greek meaning inside the head. So all the structures that are inside the cranial cavity. The image on the left is a sagittal section of a dog's head. I have marked in red the cerebrum. In yellow is the cerebellum. And in blue is the brain stem that continues through the foramen magnum as the spinal cord. The image on the center is the same, but on an MRI is a T1 enhanced sequence. You see in red the cerebrum, in yellow is the cerebellum, and in blue is the brain stem. The image on the right is the same, but I have marked different areas. This is a more clinical approach to the parts of the encephalon, also a more embryological one. In red is the prosencephalon or forebrain that includes the cerebrum and the more rostral portion of the brain stem, that is the diencephalon. In blue, we have the midbrain or mesencephalon. And in yellow, we have the rhombencephalon or hindbrain that includes the cerebellum, the pons, and the medulla oblongata. Now we perform a dorsal craniotomy and we identify inside the cranial cavity the encephalon. As you see, there is not much space between the encephalon and the bones. Both cerebral hemispheres, okay, this is the right cerebral hemisphere. Behind is the cerebellum. Dorsally, both cerebral hemispheres, the left and the right, are separated by the longitudinal fissure. So if we make dorsal sections, now we identify parts of the cerebrum, that is the hippocampus, and the lateral ventricles. And behind is the cerebellum. With another dorsal section, we see the olfactory bulbs, the rest of the cerebral hemisphere with the hippocampus here, and we have sectioned the interthalamic adhesion of the brain stem, the caudal colliculi of the mesencephalon, the cerebellum, and here we have the coded nuclei and the lateral ventricles, also here and here. Okay, we will start about the cerebrum. The cerebrum is formed by two cerebral hemispheres that are separated by the longitudinal fissure. And as you see, the more external part of the cerebrum, that is the cortex, the cerebral cortex, is folded. And between these folds, there are sulci, right? These folds are called gyri. So the cerebrum is made of gray matter and white matter. The gray matter forms the cortex or pallium and the basal nuclei. And the white matter is formed by fibers. This is a transverse section. And we see one cerebral hemisphere and the other, right? And in brown, we see the cerebral cortex, that is gray matter, and collections of nuclei included in the white matter. These are the basal nuclei. And some parts of the white matter that connect both hemispheres. We see identified here the corpus callosum and the rostral commissure. This is the same on an MRI image, okay? With the cortex, the basal nuclei, the corpus callosum here, and inside this cavity is a ventricle. These are the lateral ventricles. We also see we have cut here at the level of the optic chiasm, and here we see the two optic nerves on top of the basal sphenoid bone and the orbital fissure. We will start by the gray matter, and we'll start by the cerebral cortex or pallium. The cerebral cortex is divided in three types of cortex. One is the neocortex, the more new ones, also known as the isocortex. And then we have a transitional cortex, that is the proisocortex, and the more older one, the more old cortex, that is the older cortex, is the allocortex. This comes from the Greek meaning the older cortex. We will see that the allocortex is formed by the archicortex and the paleocortex. Okay, archi meaning the beginning, and paleo meaning old. Sometimes these two concepts are confused. So, the neocortex. This is a dorso-posterior lateral view of a dog's brain. And we see the neocortex is the more extended cortex here. You see, everything, this is the neocortex. And the neocortex is divided in different areas according to the bones that cover these areas: the frontal cortex, the parietal cortex, the occipital cortex, and the temporal cortex. We shouldn't say, as it is written in human anatomy, the frontal lobe. Why? Because in humans, in our case, or in primates, the sulci are so deep that almost 2/3 of the cortex is hidden. So this sulci can divide the cortex in lobes. This is not the case in our domestic animals. Right, and as you see here in these images, you see that the areas of the cortex are named according to the bones that are covering these cortices. So we have the frontal bone, yeah, as you see in a dorsal view, the frontal bone, the parietal bone, the temporal bone here, the scamus portion in particular, and the occipital bone. In a dorsal craniotomy, we see that the frontal bone is quite extensive, right? Then we see the scamus portion of the temporal bone and the occipital bone, right? So when we do MRIs, we identify these areas. So here is a very dorsal one, it's a most dorsal. We see the frontal bone, the parietal bone, and the interparietal process of the occipital bone. As we keep making sections, we begin to identify under the frontal bone, the frontal cortex, that is quite extended. Look at here. This, as we will see later, is the cruciate sulcus. So the frontal cortex extends beyond this cruciate sulcus. We identify the parietal cortex. And the frontal cortex is related with sensory motor functions, also related with cognitive functions, the more rostral portions. The parietal cortex has an associative function. And then as we keep going down in the cortex in more dorsal sections, we see the frontal cortex, and then it appears the temporal bone, it appears the temporal cortex, and when the occipital bone appears, we identify the occipital cortex. Right? As I want to, the occipital cortex is related with visual activities and also associative activities. The temporal cortex is related to equilibrium, hearing, and associative functions. What I want to point out is the temporal cortex is quite extended, but as well as the frontal cortex, as you see. Why so? Because in the previous image, you have identified that the frontal cortex goes quite down up to the presphenoid bone, right? So this is still frontal cortex. So as you see, the frontal one is quite a big bone, right? And extends quite down up to the presphenoid bone here. So everything of the cerebral neocortex that is covered by this portion of the frontal bone is the frontal cortex. Do not make the mistake of calling this cortex down here temporal cortex. We haven't reached the temporal bone yet. So now this is an image of a dog's head that I have performed a dorsal craniotomy. And here I have marked in yellow the parietal bone. So when we remove this bone, the cortex that is covered by this bone is the parietal cortex. Yeah. So here is the temporal bone. So this portion will be the temporal cortex. Okay. In a dorsal view, we have the parietal cortex here. And again, marked in colors, you see the frontal cortex that is quite extended. As you see, is beyond the cruciate sulcus. Okay. Cruciate sulcus here. And the parietal, occipital, and the temporal cortex. These areas are just related to the neocortex, not to the paleocortex. We have the paleocortex down here. So on the neocortex, we have, as I have said previously, gyres, okay, and sulci, and they receive specific names. In dogs, this one here is the pseudosylvian fissure, okay, and the gyre that is on top is the sylvian gyrus. Then we have the ectosylvian sulcus, and the gyrus is the ectosylvian gyrus. It's quite easy. On top of that, we have the suprasylvian sulcus and the suprasylvian gyrus. And extending from the longitudinal sulcus, a fissure, from this longitudinal fissure of the brain, we have the marginal sulcus, right, and the ectomarginal sulcus, that they are separated by the marginal sulcus. Oh, right, here, okay. And this is the ectomarginal one. In a frontal view, what do we have? We have the longitudinal fissure here and the cruciate sulcus. And the gyri that are rostral to the cruciate sulcus is the precruciate gyrus, and the one that is caudally located is the postcruciate sulcus that extends rostrally on top of the coronal sulcus. Here is an important sulcus that is the presylvian sulcus, right? That marks this area that is the prefrontal cortex, that includes the frontal gyrus and the proaerian gyrus here. The proaerian gyrus is partially separated from the frontal gyrus by the proaerian sulcus, and on the lateral aspect, separating the prefrontal cortex from the paleocortex, we have the lateral rhinal sulcus. In the cat is similar, but it's not quite similar. It has small differences. Look, again, we have the bones, the frontal bone, the parietal bone, temporal bone, occipital bone, the same. So we are going to have the same names for the neocortex. However, as you can identify quite easily here, the frontal cortex is smaller than the frontal cortex of the dog. And there you have the parietal, the occipital, and the temporal cortex is again a bit smaller. But the smallest is the frontal cortex, okay? On a mid-plane section, again, we identify sulci and gyri, and some of them are quite important. So first, we see the corpus callosum here, okay? The telencephalic septum right here. And on top of the corpus callosum, we see this is the cruciate sulcus that extends dorsal to the corpus callosum and separates the corpus callosum, or it is separated from the corpus callosum by this gyrus. That is a very important one. It's the cingulate gyrus. The cingulate gyrus extends caudally, turns around very close to the hippocampus, forming the parahippocampal gyrus, okay? And then on top of that, this sulcus here that extends from the cruciate sulcus is the splenial sulcus, right? On top of that, we have the splenial gyrus and the occipital gyrus, okay? I have marked also here these structures, the paraolfactory gyrus and the paraterminal gyrus. It is important, this gyrus, because the cingulate gyrus with the parahippocampal gyrus form part of the proisocortex. This is a transitional cortex. So remember, the proisocortex includes the cingulate gyrus and the parahippocampal gyrus. Talking about layers, we have talked about that at the development lesson, okay? But now we are going to identify in the neocortex, as we see, there are layers, up to 6 layers, okay? And you remember that the oldest layer is the more deepest layer. In this case, is the multiform layer that projects to the thalamus. And the newest is the molecular layer, right? In between them, we have other layers. That is, the outer granular layer is formed mainly by interneurons, excitatory interneurons, okay? And this layer is particularly interesting in the entorhinal cortex that we will identify later on. This entorhinal cortex projects to the hippocampus. So, and then we have a pyramidal layer, that is the outer pyramidal layer, formed by neurons that project to cortical areas that are, in particularly, they form associative and commissural fibers. So the commissural fibers come from the outer pyramidal layer, okay? Then we have an inner granular layer formed by excitatory interneurons, right? And an inner pyramidal layer. This pyramidal layer is formed by neurons that project to subcortical regions and basal nuclei, brainstem, and spinal cord. So the projection fibers are coming from the inner pyramidal layer. Remember, the associative and commissural fibers come from the outer pyramidal layer, right? And then we have the multiform layer that projects to the thalamus. The thalamus is deeply related with the inner layer of the neocortex. These layers form columns, right? So the neocortex is formed by columns, and in each column we have these layers. The proisocortex and allocortex, they also have layers, but they do not have as many layers as the neocortex. It differs, okay? And the allocortex, the archicortex, and paleocortex, we will see that they have layers. However, they do not have as much layers. Okay, for instance, the piriform cortex is just formed by 3 layers, as the olfactory cortex formed by 3 layers, as the archicortex formed by 3 layers. The basal nuclei. sometimes they are named basal ganglia. This is wrong, because a ganglion is a collection of nuclei of cells, that is of neurons, that is located out of the central nervous system. When this collection of body of neurons is located inside the central nervous system, it is a nucleus, right? And this is the case here. This is a nucleus, not a ganglion. So the basal nuclei are the coded nucleus. Here, this is the coded nucleus on an MRI here as well, right? Or here, okay. The accumbens is this part here, is located in the rostromedial portion of the coded nucleus, very in close relation with the telencephalic septum. Here is very easy to identify, the accumbens. The lentiform nucleus is formed by the pallidum and the putamen. As I have said in the development lesson, the putamen and the coded nucleus, they develop from a single nucleus that is split into portions by the internal capsule. These are projection fibers. So we have here the coded nucleus, and this is the putamen that is connected with the coded nucleus. So this is why the coded nucleus and the putamen are named striated body. And then we have, this is the pallidum here. The claustrum is located in the periphery and is separated from the putamen by the external capsule. Remember, the external capsule is formed by associative fibers, while the internal capsule is formed by projection fibers, right? The amygdaloid body. The amygdala, again, this is interesting because we have the coded nucleus and the putamen. Both together are the striated body, right? The amygdaloid body, that means an almond, right? The amygdaloid body is included in the piriform lobe. Here, the endopeduncular nucleus in carnivores corresponds to the medial globus pallidus of the primates, right? So this is to compare. This is the encephalon of a dog, and this is the encephalon of a sheep. Look, the differences between them, okay? Right. Let's have a look at transverse sections. This is a Mulligan staining from rostral to caudal. What do we see here? We see one cerebral hemisphere and the other one here. And the coded nucleus and the accumbens right here. Okay? We see the putamen. Remember that these nuclei are long nuclei, right? This is the putamen here. As we progress more caudally, we identify this coded nucleus that becomes then this classical structure that we all know. Okay? And the connections is the putamen. Again, we identify the putamen here. And the putamen, and this is the claustrum. So we are beginning to see the claustrum quite rostrally. The claustrum here, okay? And the pallidum. So these nuclei are very long structures. The telencephalic septum here, okay? And the telencephalic septum. We have got here the optic chiasm and the rostral commissure, the corpus callosum, the cingulate gyrus here, and the hypophysis. So again, if we compare it with the brain of a sheep, we see here again the coded nucleus. The coded nucleus, and here we have the putamen. These structures form the striated body, and the pallidum ventrally located again. So you see, it's similar, it's quite similar. It's not the same, but quite similar. Okay, the putamen and the coded nucleus, and here we have the pallidum located at these locations here. And as you see, the pallidum is quite long nucleus, right? The putamen is a long nucleus, more long than the pallidum, and the coded nucleus here. For a 3D image of the coded nucleus, we have to do a dissection. Here I have removed the right neocortex, the neocortex of the right cerebral hemisphere. And we have the hippocampus and the olfactory bulb that forms part of the paleocortex. Okay, we have the corpus callosum here, the cingulate gyrus, that continues with the parahippocampal gyrus. And as you see, there's a structure here that is orange-like structure. This is the coded nucleus. And here we have the lateral ventricle. And it is called a coded nucleus because it has a tail. If we look in a dorsal view, here I have removed the cerebral hemispheres. We have both thalami right here. And we see in yellow, I have marked the coded nucleus. The coded nucleus is formed by a head, a body, and a tail. This is why it is called coded nucleus. That means tail. We progress caudally, and we identify another thing, the amygdaloid body. Let's make a section here. we make a transverse section and we amplify this image. So this cortex here, that is at the level of the pseudosylvian fissure, is a cortex that is hidden because the single gyri, they have undergone an opercularization. And this is the insular cortex, an IL. This is why it is called like that. It is related with the cognitive functions and behavior. Okay, and this insular cortex is closely related with the rest of the basal nuclei, as the putamen, claustrum, and endopeduncular nucleus. This is part of the pallidum, and also this nucleus here that is hidden in the piriform lobe. This is the piriform lobe, and you know, we will see later, this is part of the paleocortex with the olfactory bulb, olfactory peduncle, and piriform lobe, and hidden inside we have the amygdaloid body. So look at the relations between the insular cortex, putamen, claustrum, and the amygdaloid body, and this is entorhinal cortex, is part of the piriform lobe. Okay, this will be a section at this level here, at this level. Right. Okay, so the basal nuclei are vascularized by the middle cerebral artery and formed by the quoted lentiform, claustrum, amygdaloid body. However, the quoted nucleus and the lentiform are related in motor functions as well as behavior. And do not forget that the quoted nucleus plus the putamen, they form the striated body or corpus striatum. So phylogenetically, these basal nuclei are divided in neostriatum, paleostriatum, and archistriatum. So the neostriatum is formed by the quoted nucleus and the putamen, and they derive from the single nucleus that evolved from a nucleus that was close to the lumen, or to the ventricle, that was split by the internal capsule. And the neostriatum is closely related with the neocortex. The paleostriatum is the pallidum, and the archistriatum is the amygdaloid body. The amygdaloid body is considered a door to the hippocampus. So this is related with the archicortex. Okay, right. What are the main functions of these basal nuclei? So these main functions are, as the basal nuclei are related with the cerebral cortex, in order to allow automatic movements, okay, and behavior. Lesions that are known as dyskinesias is a syndrome of involuntary disorder movements, okay? So that comes from the Greek wrong movements, dyskinesias. Lesions may be caused by lesions that affect these basal nuclei that are quite important in our domestic animals. However, in humans, it seems that if the lesion affects the cortex, the automatic motor functions are also disturbed, right? However, in cats, it has been pointed out that a lesion of the basal nuclei, but if the cortex it is not affected, the automatic movements are not affected. Okay. So again, how these nuclei are related to other structures. As we see, this is very simple. The motor and sensory cortex projects to the corpus striatum, and the thalamus and brainstem projects as well to the corpus striatum. The corpus striatum, in turn, projects to the pallidum. And the pallidum goes back to these areas, whether there is a motor or sensory cortex or thalamus and brainstem. So closing a circuit. And then once these connections have been arranged, then the motors and sensory cortex or thalamus can project to the brainstem and spinal cord in order to reach the muscles. So it has been pointed out that there is a direct and indirect pathway in the control of the movements. So it seems that and this is very important in order to control the right movement. What I mean is there is the direct pathway is, in this case, the cerebral cortex has excitatory projections over the corpus striatum. Then the corpus striatum projects inhibitory actions over the pallidum, the medial portion, what is known the endopeduncular nucleus. However, as this nucleus is inhibitory over the thalamus, we end up having an activation, because inhibition of an inhibition ends up in an activation. So the thalamus activates the cerebral cortex. This pathway tends to facilitate the initiation of the movement. However, the indirect way in which the cerebral cortex, it has an excitatory projection over the corpus striatum, And as we know, as in this case, the corpus striatum has an inhibitory action over the pallidum in this case, the lateral portion of the pallidum, and the lateral portion of the pallidum inhibits the subthalamic nucleus. So what is going to happen? Again, inhibition of an inhibition, we end up in an activation. So the subthalamic nucleus is activated now. So it activates the medial portion of the pallidum. And as this one is inhibitory over the thalamus, we inhibit the activity of the cerebral cortex. So a combination of these pathways, direct and indirect pathway, tends to coordinate and to allow a correct movement. One facilitates the initiation of the movement, and the other one, the indirect one, inhibits the unwanted movements. This is a summary, and it's important to notice that the cerebral cortex, as I have said, activates the striated body, or corpus striatum, and the corpus striatum projects to the pallidum. However, it may reach the medial portion or the lateral portions, and the pathways are going to be different. When reaching the medial portion is inhibitory, and the medial portion, as it is inhibitory of the thalamus, we end up having an activation. So it facilitates the movement, okay? However, when it reaches the lateral portion, in this case, as it is inhibitory, and the lateral portion is also inhibitory, we end up having an activation of the subthalamus. And the subthalamus activates the medial portion of the striatum, and as the medial portion of the striatum, of the corpus striatum, is inhibitory, in this case, we are inhibiting unwanted movements. So a coordination of these two pathways are very important, are a key for the correct movement. We have to also know that the subthalamus, it activates the substantia nigra. Remember, the substantia nigra is in the mesencephalon, and it has a pars compacta and a pars reticulata. The pars reticulata is activated by the subthalamus, and in turn, the pars reticulata has GABAergic inhibitory activity over midbrain nuclei. In this case, it controls the saccadic movements of the eye. However, the substantia nigra, in particular the pars compacta, is receiving activation from the cerebral cortex and from the striated body, as well as from the thalamus. And this pars compacta is a very important area because it projects dopaminergic inhibitory actions, dopaminergic inhibitory actions over the basal nuclei in order to control the activity of these nuclei here. So the basal nuclei are controlled by the pars compacta of the substantia nigra. Okay, so now, sorry about the slides. We have to compare the basal nuclei and the cerebellum. Why? And you will see now. Both basal nuclei and cerebellum receive activity from the cerebral cortex, both. And both basal nuclei and cerebellum project to the thalamus, and the thalamus to the cerebral cortex. So two circuits are here. One between the cerebral cortex and the basal nuclei, and the other one between the cerebral cortex and the cerebellum. What are the differences between them? So first, about the projections from the cerebral cortex. In the case of the basal nuclei, the entire cerebral cortex, okay, or the basal nuclei receive projections from the entire cerebral cortex. Okay. However, in the case of the cerebellum receives just afferences from the sensory-motor cortex. This is the first difference. Cerebellum, just sensory-motor. Basal nuclei from the entire cortex. The output. Basal nuclei are projected to premotor cortex, motor cortex, cortex that is involved in motor functions, and prefrontal associative cortex. This is formed by the by the olfactory gyrus and by the proerian gyrus. This is what is also known as the intelligence cortex, or the more cognitive and associative cortex, right? The intelligence cortex. The cortex that is in primates is very big, right? In the case of the cerebellum, the output goes to premotor cortex and motor cortex, just motor activity, not prefrontal cortex. From the brainstem, the basal nuclei receive no direct connections from the spinal cord, and some of them, but few, from the brainstem, as we have identified previously. In the case of the cerebellum, many of the connections are coming from the spinal cord, a lot of them. It's also received from the cerebellum, all the centers, and brainstem, but a lot of them are coming from the spinal cord. So, finally, in a summary, we have to say that both structures are related to motor functions. However, the basal nuclei are involved in higher order cognitive aspects of this, planning, execution, more complex functions of the motor activity. However, in the cerebellum, it just regulates the execution of the movement. These are the differences. And in this paper, the basal, this is wrong, we should say the basal nuclei and the cerebellum, okay? We see the basal ganglia, wrong, basal nuclei, the cerebellum and the cerebral cortex form an integrated network. Both are related, deeply related. So now we jump to the, allocortex or rhinencephalon, the olfactory brain, okay? Now we have studied the neocortex. Now we have to jump to this other cortex. It is also named rhinencephalon because it's deeply related to olfaction, right? And this allocortex is divided in a paleocortex and archicortex. The paleocortex with a basal portion and a septal portion, and the archicortex with the limbic portion, right? As I said before, both words mean something that is all. However, archi means beginning, and paleo means all. Sometimes these words are confused. We'll begin by the paleocortex. The paleocortex is divided in a basal portion and a septal portion. The basal portion, it is, as the word says, ventrally located. So we have an olfactory bulb, the olfactory peduncle, as you see here, and the piriform lobe. And the septal portion is a septum. What is this? Okay, look at this image here. We have a transverse section of the encephalon with one cerebral hemisphere and the other. So we have the cortex here, but as you see under the corpus callosum, these cortices from one cerebral hemisphere and from the other are in close contact, and this is a septum. And the septum has two portions. One is a cellular portion, as this one here, the cellular portion, and the other one is more dorsally located, right, between the corpus callosum and the fornix, and this is the pellucid portion. So, in the basal portion, what are the tracts, the olfactory tracts? There are three. The lateral, the medial, and the intermediate. Okay, this is a dorsal section of an encephalon, of a dog. The cortex nuclei here, right? And what do we see? We see in this green, what I have marked here, is the intermediate olfactory tract. Sorry, the intermediate olfactory tract connects both olfactory peduncles, okay? The one of the right and the left olfactory peduncles are connected through the intermediate olfactory tract. The lateral olfactory tract projects to the lateral olfactory gyrus, to the piriform lobe here, and to the olfactory tubercle. The olfactory tubercle is this structure here, located here. Remember, the cortex nucleus, the putamen, both together the striated body, the accumbens, and here we have the olfactory tract. Okay? The medial olfactory tract projects to the telencephalic septum. In this image is a, is a view of the floor of the cranial cavity after removing the encephalon. You see the oculomotor nerve, the hypophysis, the optic nerve, sorry, with the optic chiasm. And here, as we see, there are part of the olfactory bulb here. Something has remained attached to the bone. So important here to notice. Look, in this image, this is the encephalon of a dog. I can say olfactory peduncle, white matter, and olfactory bulb. Olfactory bulb, remember, is paleocortex, so it's gray matter on top. And then we see the olfactory nerves entering the olfactory bulb. However, in this image, here is the encephalon that has been removed from here. And, sorry, this is the encephalon that has been removed. And we see part of the olfactory bulb, but this is white matter because the gray matter of the olfactory bulb remained here. So it has been detached, okay? So remember that we say paleocortex because there is gray matter on top, not white matter. So when we identify that on MRIs, we see the olfactory peduncle, and on top the olfactory bulb, right? And here the olfactory peduncle. Again, we see a sagittal section of a dog's head in a dissection, right? The cruciform plate of the ethmoidal bone, right? The frontal bone with the frontal sinus, etc. And the corpus callosum right here. And what do we see? We see the structures. This structure here is the intermediate olfactory tract. And remember that this intermediate olfactory tract connects both olfactory peduncles and olfactory bulbs through the rostral commissure. Look at this rostral commissure. The rostral commissure is located at here, at a bit more rostral to the fornix. This is the fornix, right? And these fibers here are the medial olfactory tract that connects the olfactory bulb with the telencephalic septum. Here we have the telencephalic septum. So medial, intermedial, and here we lack of the lateral. So in this drawing, it's interesting to notice something. Here we have the cribriform plate of the ethmoidal bone, where all the fibers from the olfactory cells are entering the cranial cavity. Once they enter, they join in bunches of fibers in order to form the olfactory nerves. And these olfactory nerves reach the olfactory bulbs in the glomeruli, okay? From these olfactory bulbs, some of the fibers are going to form the intermediate olfactory tract that is connecting both olfactory bulbs and tracts, right? Through the rostral commissure. And we also have the medial olfactory tract that connects the olfactory bulb with the telencephalic septum. And this one is the lateral olfactory tract. Remember that this lateral olfactory tract reaches the piriform lobe. And cells from the piriform lobe are also connected with the piriform lobe of the other side, with its homologous, through the rostral commissure. So the rostral commissure has two portions. One, rostral one, that is formed by the intermediate olfactory tract, and the other one, more caudal, is formed by the fibers that connect both piriform lobes. So transverse sections, what do we see here? Okay, we see the olfactory peduncle on MRI as well, and identify we have hypointense signal from fibers and a more gray signal. This is cortex. This is why we call paleocortex. Okay, there is cortex located peripherally. Okay, again, olfactory peduncle with white matter inside. We progress more caudally, and this white matter, you see this arrow is pointing at this hypointense dot here, more black dot. This black dot, we follow that, and this is area here. This is a Mulligan staining. So we have this is the intermediate olfactory tract. Okay, this one here, and peripherally located. That is difficult to see on an MRI, but it's easy to see on a Mulligan staining or in a fixed dog. You see the white matter here. This white matter here is the lateral olfactory tract. Okay, and this lateral olfactory tract, you see, is white matter on top of gray matter, and this is a very primitive structure. Okay, it's like in the spinal cord. It's white matter on top of the gray matter. So this is a very ancient area, phylogenetically speaking. Again, we see the olfactory peduncle with the cortex on top and some white matter on top of it. Okay, right. And as you see here, this is a dorsal section, and again a dorsal section. And in this one, you see white matter located on top. This is the lateral olfactory tract. This is a very primitive structure phylogenetically, right? So again, you see the lateral olfactory tract here and the lateral olfactory tract here. And more dorsally located is the intermediate olfactory tract that is going to form the rostral commissure. Here we have the telencephalic septum right here. So what I've done here, this is a midplane section with a corpus callosum, cingulate gyrus, okay, thalamus, cerebellum, mesencephalon, pons, medulla oblongata, fourth ventricle here. I have removed the telencephalic septum and the paraolfactory and paraterminal gyri in order to discover here that this is the rostral commissure that is formed by two types of fibers. One is the caudal portion that connects the piriform lobes between them, and the other one is the rostral part, right, that connects the both olfactory bulbs. In this dissection, you will identify easily. This is the rostral commissure, and as you see, this is with the caudal portion, caudal here, and the rostral one here. Okay? So if you compare that with the encephalon of a sheep, what do you see first? What attracts your attention? The olfactory peduncles and the olfactory bulbs that are quite different from a dog. Okay, you see? Right. So now we move to the archicortex, the limbic portion. What it means, limbic, margin. To be on the limbus is to be on the periphery, on the marginal area. And it is called like that by Broca, because it's the part of the cortex that is surrounding the more rostral portion of the brainstem. Let's have a look. Here, in this dissection, I have removed the neocortex of the left cerebral hemisphere. Okay, and then you see the hippocampus as a C-shaped structure of cortex here. Here we have the piriform lobe right here. All right, and the cingulate gyrus that continues with the parahippocampal gyrus. Okay. If I made a transverse section now in an encephalon, at this point, I will be cutting the hippocampus at 2 points, dorsally and ventrally. What do we have here? We have the hippocampus section in 2 portions. Okay, we start by here, the corpus callosum, cingulate gyrus, and we continue. This is neocortex, 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 and we come to the basal portion of the paleocortex. And we continue, and this cortex folds and forms the hippocampal formation here, this one here, this one here. So in a Mulligan, in a Bielchowski, what do we see? We see this area has been enlarged here histologically. And underneath, we have this is a portion of the paleocortex that is also known entorhinal cortex. So we progress by that, and then this cortex goes up. This is why it is called subiculum. And then it becomes the hippocampus proper, this one here. And here we identify another portion of the hippocampal formation, the dentate gyrus. And as you see, all the fibers that are leaving the hippocampus right here are doing so on the periphery. Why? Because it's a very ancient structure, white matter outside the great matter. So, and it formed the alveus hippocampi. And all the fibers concentrate in one point, that is the fimbria of fornix. And the fimbria, the apex of the fimbria fornix is the taenia of fornix, where the choroidal plexuses of the lateral ventricles are attached to. Look at this structure here, and we'll identify this structure later on. But look at this. This is a tract. In particular, is the optic tract, and it's very close to the hippocampal formation. So, the hippocampal formation is formed by the subiculum, hippocampus, and dentate gyrus, right? And the hippocampus has 3 layers, and the dentate gyrus has 3 layers, right? So, how these fibers are going to get to the hippocampal formation? By ascending fibers. From where? From the parahippocampal gyrus and the caudal portion of the piriform lobe. And we know at this cortex, this cortex is also known entorhinal cortex. So there are 2 pathways, an alveus path and a perforant path. The alveus path goes up and to the hippocampus, and the perforant path goes to the hippocampus and to the dentate gyrus. And from cells from the dentate gyrus enter the hippocampus again. And all the fibers from the hippocampus, they leave the hippocampus, forming the alveus hippocampi, that these fibers are going to form the fornix, right here. Okay, so pathways from the amygdaloid body. Remember that the amygdaloid body is a basal nucleus on base, right? Basal nucleus, one of them. And it is also known as the door to the hippocampus, one door. These fibers are going from the amygdaloid body, entering through the parahippocampal gyrus. This is the entorhinal cortex, and entering to the hippocampus. So let's understand this hippocampus and the fornix, okay? These fornix are the fornix are the fibers that are leaving the hippocampus, and they form an arch that surround the thalamus, okay? So the fibers that leave the hippocampus, they form the crus of fornix. They form the crus of fornix right here, and they go up. Some of the from both fornices, the fibers are connected by the commissure of the fornix. Here there is a ventral view. Ventral view. You see the hippocampus here. All the fibers are leaving, forming the crus of fornix. The crus of fornix. Some fibers are connected rostral to the hippocampus. They form the commissure of the fornix, and then the fibers go rostrally. They meet at the midplane, forming the body of the fornix, and then they split again. They split again, forming the columns of the fornix in order to reach hypothalamic nuclei. These are the mammillary nucleus or mammillary bodies, right? Right, this is wrong. This should be the body of fornix should be located here, not here. This is not the body of fornix. All right. Right. So let's understand that. What do you have? This is a ventral view of the encephalon, and we make a longitudinal section, right? I have removed 1/2 of the cerebrum, and there is not thalamus here, not brainstem, not thalamus. So we see the olfactory peduncle, the piriform lobe here, that extends caudally, forming the parahippocampal gyrus and continues with the cingulate gyrus. And this area, marked in yellow, is the entorhinal cortex. So now we make a section and we split this. We remove this part of the cerebrum in order to identify, as you see, the parahippocampal gyrus here, as it goes, folds and forms the hippocampus and traps this area here that is the dentate gyrus. Maybe you will understand better in this image. Again, the parahippocampal gyrus, here is this one here, that goes up, forms the subiculum, the hippocampus proper, and here it is the dentate gyrus because it has dentations. Okay, and all the fibers go out from the hippocampus, forming the alveus hippocampi, and all these fibers join to form the fornix. Look at the lateral ventricle here. So on MRIs, when you see these sagittal sections, the lateral ventricle, the cingulate gyrus, parahippocampal gyrus, folds and forms the hippocampus. This should be the subiculum and the hippocampus. And this one is easier. The cingulate gyrus continues with the parahippocampal gyrus, okay, with the parahippocampal gyrus, and then goes to the hippocampus. As this is a midplane section, as this one, we do not see anymore the hippocampus. What do we see is the dentate gyrus that is more medially located, extends medially. This is the dentate gyrus, right? As you see, the cingulate, parahippocampal gyrus, and we miss the parahippocampal gyrus because it goes laterally. Now we have the dentate gyrus, right? And these are the fibers that are leaving the hippocampus. They form the fornix. At this level, we have the columns of the fornix running down and caudally to the rostral commissure. This is the optic chiasm and the hypophysis. And as you see on an MRI, this is the corpus callosum, cingulate gyrus, parahippocampal gyrus, and we miss the parahippocampal gyrus now, but we identify the dentate gyrus and the fibers of the fornix right here. Again, this is the lateral ventricle and the hippocampus, the hippocampal formation. And as you see, this alveus hippocampi is forming part of the wall of the lateral ventricle. This is the lateral ventricle and the amygdaloid body here. Again, you see the parahippocampal gyrus and continues and folds and form the hippocampal formation here. And as you see, parahippocampal gyrus and hippocampal formation. This is the same. Cingulate gyrus, parahippocampal gyrus that goes laterally and is missing here, but we identify the dentate gyrus and the fornix, okay? The fornix here. So again, because it's a complicated structure, we make a dorsal section here, and also we have removed the paleocortex and the neocortex, but both hippocampal or archicortices remain, okay? So the hippocampus, and also we see that we have cut at this level the corpus callosum at the genu, or rostrum, genu, and splenium of the corpus callosum. So the fibers that leave the hippocampus, this is why it is white, because we have white matter here. This is covered by white matter. It's a very old structure phylogenetically. So the fibers convert and form the crusofornix. They join the commissure of the fornix, they join and they form the body of the fornix that is related with the telencephalic septum, sorry. And we go back here. What do we see? Now we have moved both hippocampi to the left. And we discover that the both thalami, this is why it is, this cortex is limbic cortex, because it forms the limbus, okay? The margin, the cortex of the margin that covers the thalami, or the rostral portion of the brain stem. So you see, again, hippocampus, crusofornix, they go up, they go rostrally, the body of the fornix, and this structure, that is gray matter, is not anymore white matter, it's gray matter. This is the telencephalic septum, this one here. As you see, the lateral ventricles, and here we have the third ventricle, and the foramina that communicates the third ventricle with the lateral ventricle, right? And the coded nucleus with the tail, the head, the body, and the tail, right? So now we have moved the hippocampi rostrally, and we identify the ventral portion of the hippocampal formation with the dentate gyri, both, and the fornix. This is the fornix with the commissure of the fornix, and they go up columns of the fornix. And you see, when we push rostrally the fornix, we clearly identify the columns of the fornix. In these images, here is a dorsal view. The part of the cerebral hemispheres have been removed, the neocortex and the paleocortex. However, the archicortex is not removed, it's still there. So we have these horns, the asta ammonis, right? And we have the fibers, this is white matter covering, this is white matter, and all the fibers that leave from the crusofornix, the commissure of the fornix, the body of the fornix, right? In this 3D image, we see the cingulate gyrus continues with the parahippocampal gyrus and is missed because it goes laterally. The dentate gyrus, the interthalamic adhesion, the mesencephalon, the pons, the medulla oblongata, the fourth ventricle with the choroidal plexuses of the fourth ventricle. So, and the corpus callosum. And underneath we see the fornix. So look at the relation between the fornix and the corpus callosum, right? Here. So now we can identify in Mulligan. We can identify many structures. the coded nuclei, right? The telencephalic septum. And now we know that this structure, that is white matter, that it goes ventrally and passes caudal to the rostral commissure, is the columns of the fornix. And they have to reach here, the mammillary bodies, right here. This white point are the columns of the fornix that reach the mammillary bodies of the hypothalamus. And more caudally, at the level of the metathalamus, the section, this is the substantia nigra, right, of the mesencephalon. What do we see again? We see the cortex is neocortex. Now we come to the paleocortex, entorhinal cortex, goes up and falls and forms the hippocampal formation. And on top we have white matter, the alveus hippocampi, that is going to form all these fornix, right here. We have these fornix, and you see is white matter on top of gray matter. And in this one now is a more caudal one at the level of mesencephalon and part of the pons and part of the thalamus with the lateral geniculate body and the medial geniculate body. And now you will understand perfectly well how this cortex is going around the thalamus. This is why it is limbic cortex, is the limbus, right? And this is a case. And what I want to point out in this case is this lesion here, right? And this lesion is affecting what? Which structures? This structure here is part of the hippocampal formation. However, if we know anatomy, we know that this hippocampal formation is very close to this optic tract, okay? So the optic tract in this case is also damaged. So this animal may have signs of misbehavior, etc., but also visual problems because the optic tract is affected. Here we are going to talk about the white matter. The white matter is formed by fibers. And there are three types of fibers: commissural, association, and projection. Commissural fibers are fibers that communicate both cerebral hemispheres. And there are three: commissure of the fornix, rostral commissure, and the corpus callosum. The commissure of the fornix communicates both fornices. So it belongs to the archicortex. Right? You have it here in this drawing. These are the fornix, parts of the fornix, and then the commissure of the fornix. The rostral commissure is paleocortex, communicates the olfactory bulbs and also the piriform lobes. And finally, here is in green, and finally the corpus callosum. The corpus callosum communicates the neocortices, right, between both hemispheres. And you have it here, the corpus callosum, and here the corpus callosum. Association fibers are fibers that communicate different areas, but inside of one cerebral hemisphere. They do not cross, or they do not project to different areas apart from the cerebrum, right? The projection fibers are fibers that project outside the cerebrum, or they get to the cerebrum. And here we have the internal capsule. We begin by talking about the corpus callosum. Corpus callosum comes from the Latin meaning hard body. The corpus callosum communicates the neocortices of both cerebral hemispheres. Here is a dorsal view. I have removed the parts of the gyri that are close to the longitudinal fissure, the marginal gyrus and the ectomarginal gyrus of both cerebral hemispheres. And we see the fibers that communicate the corpus callosum. Again, the corpus callosum between both cerebral hemispheres under the cingulate gyrus. Here is a mulligan staining, and we identify the corpus callosum. The corpus callosum has different parts. One is the rostrum. The rostrum communicates motor cortices, primary and supplementary motor cortices. Then we have the genu that communicates the prefrontal cortex between both hemispheres. Then we have an extended body of the corpus callosum that communicates motor and some aesthetic cortex. And then finally we have the splenium that communicates temporal and occipital cortices of both hemispheres. This is a dorsal section of both hemispheres, cerebral hemispheres. You see the coded nucleus with a long tail, right? The head, the body, and the tail. And here we have corpus callosum, right? The rostral commissure. The rostral commissure, as we have explained before, communicates the olfactory bulbs. Where is this commissure located? Here. It is located just rostral to the columns of the fornix, right here. Right here is the rostral commissure. You see the rostral commissure. And the rostral commissure is located at the same level but dorsally to the optic chiasm, because both are originated from the lamina terminalis that extends in the embryo, the rostral end of the diencephalic vesicle. And as you see here, this is the rostral commissure, and behind we have the columns of the fornix. Okay, so remember that the rostral commissure just dorsal to the optic chiasm. Let's have a video. Now we split both cerebral hemispheres because I have cut the corpus callosum. And this is just to identify here the rostral commissure. This is the rostral commissure here. Right? It's quite easy to identify in a dissection, the rostral commissure. Okay, so now we go to the fornix. The fornix, that means from Latin volt. Okay, as we have said previously, they are the fibers that are leaving the hippocampus and they form a bolt around the thalamus. So it's formed. So the fornix is formed by commissural and association fibers. Association fibers are fibers that communicate different structures inside one hemisphere. They do not cross to the other side. So these are fibers that are coming from one hippocampus. They go up, they go rostral, they form the body, and then the columns of the fornix that reach the mammillary bodies of the hypothalamus. However, there are some fibers that communicate both crucial fornix, and these are the commissural fibers. So here you see the corpus callosum and the fibers of the columns of the fornix going rostral and down in order to reach the mammillary bodies, passing just caudal to the rostral commissure. There you have that, another dissection where you see the crucial fornix going up and being related with the telencephalic septum here. Okay. Now this is a dorsal view of an encephalon. It's covered by the meninges. The cerebellum behind, the cisterna magna. And underneath we identify the hypophysis. Okay, I have removed the meninges. This is the longitudinal feature, right? Okay. All right. With all the gyri, the occipital cortex, the cerebellum, and the cisterna magna there. You see, this is the cisterna magna. This encephalon has no dura, but has arachnoid. So this, we have the hypophysis and the optic nerves with the optic chiasm, and the internal carotid arteries. You see the olfactory bulb here, and continues caudally as the olfactory peduncle, and this sulcus is the lateral rhinal sulcus that separates the neocortex from the basal portion of the paleocortex. And what I want you to show here is here you see this is the piriform lobe, is entorhinal cortex, continues with the parahippocampal gyrus. This is entorhinal cortex. This is the entorhinal cortex. Now we separate both cerebral hemispheres and we identify the corpus callosum. Well, right, with the rostrum, the body, and the splenium, and on top the cingulate gyrus. See, this is the corpus callosum and splenium of the corpus callosum. Now we remove part of the neocortex in order to identify here is the insula, the insular cortex. This is the piriform lobe that extends caudally as the parahippocampal gyrus, the white matter. The insular cortex, or insula. Having removed most of the neocortex, we have the transition cortex, that is the cingulate gyrus, on top of the corpus callosum. Right. We remove the cingulate gyrus and we identify here the corpus callosum. Commissural fibers. Okay, as you see, the cingulate gyrus continues caudally as the parahippocampal gyrus and continues as the piriform lobe. And this portion, the caudal portion of the piriform lobe and the parahippocampal gyrus are known as the entorhinal cortex. Fibers from the entorhinal cortex reach the hippocampal formation. Can you see the corpus callosum there? And the amygdaloid body. This is the amygdaloid body inside the piriform lobe. This is the piriform lobe, right? So now we identify the fornix. This is the fornix, the corpus callosum, and underneath here we have the telencephalic septum with the pellucid and cellular portions. Telencephalic septum, you see how it is closely related with the fornix. Right, this is a dorsal section in order to now to focus on the internal capsule. Okay, the cerebellum behind, right? The caudate nucleus, right here. Okay, the hippocampal formation, the parahippocampal gyrus here, parahippocampal gyrus, both thalami. And then you see this is the internal capsule. The internal capsule is divided in three portions. The rostral crus here, the genu and the caudal crus. The rostral crus is between the caudate nucleus and the lenticular nucleus. The caudal crus is between the thalamus and the lenticular nucleus. This is the lenticular nucleus. And the genu is in between. So the rostral and the caudal. And in the caudal crus, we have the optic radiation. Let's have a look at the optic radiation. In this dissection, I have removed part of the neocortex in order to identify this. The optic chiasm with the optic nerves, the optic tract, fibers that are coming from the optic chiasm until the lateral geniculate body of the thalamus. And fibers from this lateral geniculate body, they leave and they form the optic radiation that projects to the occipital cortex. Let's have a look. You see the optic tract here, the crus cerebri, right? And the optic radiation. This is the optic radiation. And you see the lateral geniculate body here. And these are optic fibers, right? So remember, this is the optic radiation that is part of the caudal crus of the internal capsule. So if we make a transverse section of the encephalon, here is a Mulligan staining. And what do we have? And we are going to focus on the white matter. We see the corpus callosum, these commissural fibers, the rostral commissure, commissural fibers, ¿vale? Fornix, association fibers, right? The internal capsule, projection fibers. And when the corpus callosum that is formed by commissural fibers meets the internal capsule, then we have the centrum semiovale. Fibers from this centrum semiovale, some of the fibers may project to the cortex. They form the corona radiata. However, we also have fibers that are longitudinally. So fibers that connect different areas, association fibers. Some of them perpendicular, some of them longitudinal. So what are they? These are the external capsule and the extreme capsule. The external capsule is between the putamen and the claustrum. And the extreme capsule is between cortex and claustrum. And these are associative fibers. Also, there is this subcallosal fasciculus here located. Okay, because it's located underneath the corpus callosum, subcallosal fasciculus. And this is a fasciculus that connects frontal cortex with occipital cortex. We also have here at this level the superior longitudinal fasciculus. It's a fasciculus formed by associative fibers that connect the frontal cortex with the parietal cortex with the occipital cortex. We also have the uncinate fascicle. This fasciculus connects the temporal cortex with the frontal cortex, or the inferior longitudinal fasciculus that connects the occipital cortex with the temporal cortex. In these enlarged images, this is a Bialchowski or Mulligan. This is an impregnation, and this is a staining, right? So we have the caudate nucleus, the internal capsule with the pallidum and putamen and claustrum, internal capsule with the putamen and claustrum, right? And this number two here is the superior longitudinal fasciculus. This fasciculus is located just on top of the putamen, intermingled with the, or mixed with the fibers of the internal capsule. And here, just very medial to the centrum semiovale and at the dorsal to the most lateral portion of the coated nucleus is the subcallosal fasciculus. It's a fronto-occipital fasciculus, right? Because here we have the corpus callosum. Okay? So this fasciculus is located just top of the lateral aspect of the coated nucleus. This is the tail of the coated nucleus. So now we are performing a dissection with the technique of fiber dissection. We remove part of the neocortex, the sylvian gyrus, suprasylvian gyrus, and we identify all these fibers that form the internal capsule. Okay, we keep removing cortex and white matter. And we discover the superior longitudinal fasciculus here, these fibers that connect the occipital cortex with the frontal cortex. We keep removing cortex with internal capsule. And we reach the centrum semiovale here. You see all these fibers of the superior longitudinal fasciculus. Remember that the centrum semiovale is when the internal capsule meets the corpus callosum. It's this area here. Okay, we keep dissecting with this technique. In order to achieve these images of the fibers, the encephalons have to be frozen and defrozen several times. So you see, these fibers here belong to olfactory tracts. These fibers belong to the fornix, and these fibers belong to the optic tract. And then we have the centrum semiovale. You see, the optic tract coming from the optic chiasm and going up to the lateral geniculate body. You see the corpus callosum here that joins the internal capsule and forms the centrum semiovale. It's this area. Now we discover the coated nucleus here. This is the coated nucleus, and we introduce the forceps in the lateral ventricle. The centrum semiovale again. And now we have cut through the centrum semiovale, and we identify the lateral ventricle here. You see, this is the coated nucleus, the corpus callosum, and we see the lateral ventricle. This is the lateral ventricle here. Now we proceed the fiber dissection, and we go to the hemisphere on the right, but we are going from medial to lateral, right? We identify here the coated nucleus, right? This is the coated nucleus. This is a medial vision with the tail of the coated nucleus and the head. The lateral ventricle here. So the coated nucleus, you see, is gray matter. we remove part of the coded nucleus and we identify what is lateral to the coded nucleus, the internal capsule. This is the tail of the coded nucleus. And lateral, lateral to the coded nucleus, we have the putamen, the lenticular nucleus. And here we also have the lenticula, the pallidum. So you see, the lenticular nucleus is lateral to the internal capsule. These are fibers from the internal capsule. This is another dissection. This is a dorsal view with the longitudinal fissure. And we split the encephalon through the midplane. And we identify the corpus callosum, right? The interthalamic adhesion, mesencephalon, pons, medulla oblongata, cerebellum, right? Cerebral hemisphere, right? The fornix, the telencephalic septum with the pellucid portion and the cellular portion, paraterminal gyrus. Okay, you see the pellucid portion here and the cellular portion of the telencephalic septum. We remove the gyri from medial to lateral now again. The corpus callosum here. And now we have removed the cingulate gyrus with the part of the parahippocampal gyrus. And as you see, these fibers are fibers from the corpus callosum, and these are fibers from the corona radiata. Okay. We have removed the splenial gyrus. And we identify here the dentate gyrus. This is the mesencephalic aqueduct and mesencephalon. From a caudal view, the mesencephalic aqueduct here, the fourth ventricle, and the parahippocampal gyrus has been removed. Now the brainstem has been cut transversally at the level of the mesencephalon pons. Okay. As you see, the parahippocampal gyrus was continuous with the piriform lobe. Now the thalamus has been removed. And what do we see from medial to lateral, from the inside? The splenium of the corpus callosum, right? And here we see the hippocampal formation. Together with the fornix. This is part of the fornix here. Okay, this is part of the fornix. And more externally, forming part of the lateral ventricle, the walls of the lateral ventricle, the hippocampus. Now we push the hippocampus out, the hippocampus, right? And this is the lateral ventricle. You see how the lateral ventricle has a C shape as the hippocampus and the fornix. Here again. And when we remove the hippocampus and fornix, we clearly identify the lateral ventricle. And another wall of the lateral ventricle is the coded nucleus with its tail. So you see the coded nucleus here and the tail of the coded nucleus. So if we compare the anatomy of our domestic animals, the dog, cat, you see the cat is smaller. We have seen it before. The pig with the long cruciate sulcus is directed dorsally, not as in the case of a dog. Or in the sheep with a short cruciate sulcus. Or in the horse. The horse has an ovoid shape and the cruciate sulcus is only visible in the medial plane. Or in the ox, pigmented. So in the sheep is also pigmented. Well, thank you very much.